me close that. There we go. Thank you all for coming, and thank you for Ruth for organizing this, of course. And this is I think all of you, or if not a vast majority of you, have wrote letters of support for this project that just recently got funded, that we're very excited about through the Environmental Protection Agency. It was a grant to myself, to Dr. Carmichael and Dr. Sinead Hein as well. And so we're going to, run through a bit of the overview of the project, the objectives of the project, some of our approaches, the sites that we're aiming to look at, and then of course open this up to you guys to give us some feedback. There's a little bit of wiggle room in there that because we want to definitely work with our partners. And we want our partners to have, some input, but of course we are obligated to, what we wrote in the proposal. So, and, of course, obligated to the budget. But of course, there's going to be some, room to add more and to cooperate and things like that. So that's what we're excited to bring you guys together today for. And this will be the first of, a few more meetings throughout this project. So we really appreciate it. So thank you. I'm going to start off by introducing myself, and then of course Dr. Carmichael and Sinead Hein is going to introduce herself, Sinead, and then we're going to go through and have each of you I've listed the partners there alphabetically. And we're going to have each of you perhaps just tell us your name and your title, and we'll keep that part brief. Then we're going to, like I said, going to go over the overview of the project and some of our approaches that we have proposed, and of course how we can work with all of you to either add to those approaches or get some feedback from you all. And first of all, as you saw, the meeting is being recorded. We had some partners that weren't able to attend. If you have any questions, first of all, I ask that your microphones are off in case we have any, outside noise. We're all gathering from different places. If you have any questions you can utilize the chat. We'll take a look at the chat and, of course, monitor that. Also, we like the little hand raise icon. That's also great to use. And, so we definitely want you all to participate and have lots of questions, I would assume. All right, let's see. So first of all, I'm Brandi Keese. I'm an associate professor at University of South Alabama and a senior marine scientist at Dauphin Island Sea Lab with Dr. Carmichael. And I'm a microbial ecologist. I'm a microbiologist by training. I've worked a lot in the deep ocean and the deep subsurface, but I've also worked in the Gulf of Mexico hypoxic zone. I really try to understand how microbes influence biogeochemical cycles, and also and try to understand how microbes interact with other microbes. I do that from the genetics standpoint. So my work revolves around molecular microbiology. So DNA, RNA, metagenomics, metatranscriptomics. And in this context, I find it interesting to apply those microbiological omics techniques, the metagenomics and metatranscriptomics, to problems like we're facing here and that we're going to talk about with our more microbial source tracking. And I'm going to turn this over to Dr. Carmichael to introduce herself.
I think I know almost everybody. I am Ruth Carmichael. I'm a senior marine scientist at the Dauphin Island Sea Lab and professor of marine sciences at the University of South Alabama. My work, as most of you know, focuses on inverts to marine mammals. And largely, I think the best descriptor of my work in recent years has really become sort of disturbance ecology, but it's basically, trying to understand how things people do on land and the water ultimately affect the habitat, the system, and then how animals are responding to that. And a big portion of that for my entire career and starting out even when I was in graduate school, has focused on wastewater and how human wastewater, or a combination of wastewater from human and wildlife sources, and then associated contaminants or pathogens, are affecting water quality. And a lot of my work has, well, I've focused on outcomes for animals, like, how do shellfish respond? How do, other types of invertebrates or mammals respond? A lot of my work has really taken the track of trying to understand indicators. So understanding what can we look at in the environment to tell us whether there's a human source that's a problem. And that's really, I'm really excited about this project because I feel like this really is squarely in my wheelhouse, but I get to work with cool people like Dr. Reese and Dr. Sinead Hein. I'll work on it because they do really cool techniques that are above and beyond my capabilities. And I'm really excited about putting together our multiple different approaches for what we can do for hopefully all of you. So thanks. And I'll turn it over to Sinead.
Hi, everyone. I'm Sinead Hein. So I'm an associate professor in the biology department at South Alabama. And so I'm another microbial ecologist or environmental microbiologist. And the work in my lab focuses on how microbes interact with the environment. And so I do things like study oil biodegradation, how microbes deal with toxic metals like mercury, things like arsenic, and do some work on microbial source tracking and doing things like looking at fecal indicator bacteria. So interactions between microbes and the environment, going from genes to the proteins and enzymes they use to do the work, to looking at genomes and transcriptomes and so on. And I'll leave it at that. I'll point you to my webpage if you want more.
Thank you. And honestly, this proposal is very much a team effort. My name might happen to come first, but it's certainly, we're all very, equal partners on this in the writing and the formulation of these ideas. So, and so this is really special that we could bring all of you guys together on this. I listed the partners here alphabetically. I'm just going to go ahead and list through them. And if you are a part of that organization, if you could just unmute yourself, let us know your name, your title, or your role in that organization. So Alabama Association of Conservation Districts.
Good morning, this is Casey Fulford. I am the Coastal Restoration Planner for AACD, and I work in both Mobile and Baldwin County.
Great. Is there anyone else from AACD here? Okay. All right, Alabama. Oh, sorry, go ahead. Just you.
No, I just want to say just me, sorry.
That's right. Alabama Department of Conservation, Marine Resources Division.
So good morning, everyone. I'm Scott Bannon. I'm the division director for the Marine Resources Division. So I'm a knuckle dragging enforcement officer by trade. So all those big words that Dr. Reese used, I don't even know if I can go back and Google them. So I'm along for the ride. I probably create a little wastewater along the way, but I appreciate everybody's efforts here. And we always use so much stuff that comes out of your all's projects and things. So again, thank you guys for that. And I'm not sure if any of our other staff is actually on here today, but I'll be quiet and let them announce themselves.
Okay, Alabama Department of Environmental Management.
Good morning. This is Fred Leslie. I'm the chief of the Montgomery branch of Field Operations Division and also the Water Quality Monitoring Coordinator for the department.
Fantastic. Thank you. Anyone else from ADEM? Okay. So we have Alabama Marine Mammal Stranding Network. So see Dr. Bloodgood here and Elizabeth.
Hi everyone, I'm Jenny Bloodgood. I'm the veterinarian for the Stranding Network, and I'm also a postdoctoral researcher under Dr. Carmichael.
Hi everyone. I hope you can hear me. I was having some microphone troubles. I'm Elizabeth Heeb. I am the manager of the Manatee Sighting Network at the Dauphin Island Sea Lab and a responder with the Alabama Marine Mammal Stranding Network.
Auburn University Shellfish Lab.
Good morning. This is Scott Reichard. I'm the facility and hatchery manager at the Shellfish Lab. We also have 2 open water farm sites where we raise shellfish in Portersville Bay and Grand Bay. So that's our interest here in some of this work.
City of Mobile. Oh, sorry. Go ahead.
Yeah, Rusty Grice. I'm both with the Auburn University Shellfish Lab and the Mississippi Alabama Sea Grant Consortium, and I'm an extension specialist supporting commercial oyster farmers in Mississippi and Alabama.
Thank you, Rusty. Anyone else from?
By the way, I'm the Chief Resilience Officer for the City of Mobile.
Fantastic. Anyone else here from the City of Mobile? We have Dog River Clearwater Revival, which I love that name, by the way.
Thank you. Good morning. I'm Morgan Counts and the Executive Director of Dog River Clearwater Revival.
Thank you. Anyone else here from Dog River Clearwater Revival?
Yeah, I'm Mamie Farron, and I'm sort of an advisor for Dog River Clearwater Revival. I'm retired from the University of South Alabama.
Oh, fantastic. FDA Gulf Coast Seafood Laboratory.
Hi, this is Bill Burkhart. I am the director of FDA's Gulf Coast Seafood Laboratory located on Dauphin Island.
Fantastic.
Kevin Kelty, research microbiologist at FDA.
And I'm Jessica Jones. I'm the microbial hazards branch chief at the GCSL.
Great to have you. Thank you. Anyone else? All right, Grand Bay National Estuary and Research Reserve.
Hi, I'm Kim Cressman. I'm the, sorry, Margot. There's 2 of us. I'm Kim Cressman. I'm the research coordinator at Grand Bay NEAR.
And I'm Margaret Poston. I'm the coastal training coordinator for the Grand Bay NEAR. And hopefully I can take the science that you guys have and put it in the hands of the people who can use it. So thank you.
I'm Amanda Free, and I'm doing a co-op with the NEAR, the Grand Bay NEAR, and Mississippi State. I'm a grad student.
Yes, good to see you again, Amanda. Thank you.
Yes, good to see you.
Mississippi Alabama Sea Grant Consortium.
Yeah, Eric Sparks. I'm with Mississippi State University and also Mississippi Alabama Sea Grant Consortium. I'm kind of like the catch-all coastal ecology specialist for Sea Grant. It's like everybody has their defined fields, and then it's just like whatever else. Eric can figure that out, right? So microbial source track has been something that we're working with the NEAR on now. So excited to be here.
Fantastic.
Good morning. I'm LaDon Swan. I'm the director for the Mississippi Alabama Sea Grant Consortium, and I appreciate this project being funded. We have a project that's looking at something very similar to what you're doing through the University of Alabama, Dr. Patty Sebecki. It's in its 2nd year. Its focus is only on source tracking in the Fowl River area. And this is due to the closures that Alabama Department of Public Health did based on counts and really couldn't distinguish what the source was. So that's our interest.
Excellent. And, and that brings to mind, if there's other partners that we need to, mention, or we need to bring into this, or we need to discuss, join forces with. I think that's great to mention. So thank you for mentioning Patty Sebecki. I knew her when I was a grad student a number of years ago. So that's great to know. Let's see, now we've got, we said Sea Grant Consortium, yes. So Mobile Bay National Estuary Program. Oh, Melissa, I think we're there. Try again. I can't. Can anyone else hear Melissa? Okay. Yep. Do you want us to come back to you? Okay, we'll come back to you. All right, is there anyone else here from Mobile Bay NEP? Okay, all right, Mobile Baykeeper.
Hi, my name is Cassie Bates. I'm the program coordinator at Mobile Baykeeper.
I haven't yet been here for a full year, but I'm super excited to partner and help with however we can moving forward with this project.
Fantastic, thank you. Navy Cove Oyster Company.
So Chuck said that he was going to call in, but he, oh, there you go.
I'm here, Ruth. I'm sorry. I had to star 6 to unmute. Yeah, this is Chuck Wilson with Navy Cove Oysters. Certainly know many of you from my day job with Gulf of Mexico Research Initiative and other historic parts of my life. But we are certainly appreciative of being included, and we're very concerned about the water quality in Mobile Bay. Our farm is at the in Fort Morgan, about 3 miles from the ferry landing, so we get a lot of tidal flushing that comes down Weeks Bay area, the intercoastal waterway, and then in and out of Mobile Bay, and then we're certainly influenced by Mobile River. So we're very interested in source tracking and maintaining a safe and sustainable seafood industry. Thank you.
Thank you so much for being here. And Weeks Bay National Estuarine Research Reserve.
Hey, good morning, everyone. I'm Mike Shelton. I am the Training and Watershed Program Coordinator here at the Weeks Bay Reserve. Of course, we're part of the Alabama Department of Conservation and Natural Resources Lands Division Coastal Section. And I have conducted microbial source tracking work in Fish River in the past, and looking to build on those past studies and increase the ability to help try to manage these microbial inputs into the river. We do have a TMDL on Fish River and trying to meet those reduction criteria.
Thank you. I'm going to come back to, oh, is there anyone else here from Weeks Bay? Okay, I'm going to come back to Melissa, Mobile Bay National Estuary Program. Oh, no.
Can you hear me now?
Yes, yes, great.
Okay, so I was like called in and my audio wasn't picking up and it said I was unmuted. Anyway, Zoom. So I'm Missy Partika. I'm with Mobile Bay NEP and Auburn University. I also happen to be associated with Mississippi Alabama Sea Grant Consortium. So I'm an extension specialist with Auburn University. I'm the science and monitoring lead at the NEP, and so that's the hat I'm wearing today. But my background is in coastal ecology and waterborne zoonotic diseases. So I'm interested in hearing about the work you guys are doing.
Have I left anyone out?
I think we have two people. Dr. Carl Cloyd and Ronald Bond. Carl, why don't you introduce yourself first?
Hi, I'm Carl Cloyd. I am a trophic and movement ecologist at Dauphin Island Sea Lab, and I work with Dr. Ruth Carmichael.
And is Ronald on? Okay, there maybe he popped off. That's all I see. Is there anyone else that we missed?
And did we miss any organizations that weren't listed? Okay, good. And Ronald is with what organization?
I have no idea who Ronald is. So
Ronnie he's actually my husband. So he's with the University of California, Davis, but he's a water quality specialist, and he does a lot of sampling in and around oyster growing areas. And so I thought he might be interested, and he goes, I'm not a stakeholder. So he bugged out.
Oh, well,
he
definitely
is a stakeholder.
Definitely welcome. Yes, he is.
Well, that's what I said. So I might convince him to come back on so he can hear what you guys are up to.
And informed people and people that want to know about it, I think is yeah, absolutely. It's not a closed group. That's for sure. So if he decides to join, that'd be great. All right, so I'm going to go just, a little bit of background. Everyone here is familiar with microbial source tracking a little bit. Fecal microbes are very common in U.S. surface waters. It's not just human, and that's always a big surprise. We are very fixated on, the human contribution to, these sources of feces, but it actually is, a majority of poultry, cattle, swine, wildlife, including boar. Birds are into that, in there as well, but, they're on the order of, very small percentage. Human is only about 0.1%. And this is according to, data from 2000. But there's over 10,000 water bodies that have been impaired by fecal pollution. So there's an estimate was that 1 billion tons per year of fecal material are produced in the US. As Ruth helped us convert that to 3 million Boeing 747 jets or 9 million blue whales in equivalent waste. That's just to kind of wrap your brain around that. And a lot of that can go unnoticed. And of course, but what doesn't go unnoticed is the microbial degradation of that microbial impact that has. And that's, we can, that does have implications for public, economic, and ecological health. A lot of our current water quality tools are based on total fecal pollution. They're typically non-specific indicators. These are what we call our classical microbiology tools, and they typically don't discriminate among sources. We just get a total load. And there is the assumption that all bacteria is bad bacteria. And we know that there's quite a bit of it that is normal, that's natural, that is benign, that's needed to break down that carbon and to recycle those that fecal sources. But there are, of course, some that do impact health and environmental quality. And that's what we need to try to understand and wrap our head around. There were 4 objectives in this proposal, and so I'm going to outline the 4 objectives here. I'm going to start with one, and then I'm going to let my co-PIs to discuss some of the others as well. The first one was that we wanted to define the sources of microbial sources to Alabama water, specifically Mobile Bay, and our goal is to distinguish, wastewater treatment plant from septic, from wildlife, those birds and boars, livestock, and other non-human inputs. And we're going to do this in 3 ways, right? Capitalizing on each of our strengths of the researchers here, and of course, bring you guys into this by linking to your data sets and linking to your tools, and also we want to make the most comparative data sets that we possibly can by using classical microbial indicators, using the methods that you might be using in your labs that we've traditionally been used, that has been used in the literature and in the data sets that exist. And that includes, fecal coliform, male-specific coliphage, E. coli. There's biogeochemical indicators that are very important as well, nutrients, and then, of course, the isotopes of C13 and N15, and that can really help us discern where these bodies of water are coming from. And we're going to use some more advanced microbial source tracking approaches, some that are that have been you might be familiar with already. That's quantitative PCR. That just means we're going to target, specific bacteria that have a specific gene that we can, associate with a particular organism. And then another one called eDNA, which is environmental DNA. We'll talk about that a little bit more at the end, but in general, that is all the DNA that is in our sample, in our water sample. And if you sequence, meaning get your A's, T's, C's, and G's, and you get enough of that, then you can start to discern in relative, amounts of what organisms are contributing to that into that particular sample. So qPCR, as it sounds, is quantitative, and eDNA, if we can link that together, we can get a better picture of, who is there, and then also in what amounts. And then, of course, then adding that all together, which brings us at that confluence, right there in the middle of, linking these data sets, bringing in different types of data sets, and really trying to, discern both historical and future data types, and just bring it all together. I'm going to turn this over to Sinead.
Okay, and so so we're basically going to pull all those tools together to try and define the conditions, the location, the timing of these different indicator microbes, right? So we're going to have initial effort in the project is going to be to collect and collate and examine the data that have been collected by, some of our labs, some of the stakeholders in the meeting today, and other groups across the region. And moving forward as we go into our data collection effort to make links between those past data sets, local environmental conditions, some examples here of those kinds of data. But to try and use this project as an opportunity to pull everything together and take a really kind of bird's eye view of all the potential measurements we could take. There's been a lot of work done, I think, in the past where people have collected really good FIB or fecal indicator bacteria data sets. In my lab, we've collected some qPCR type microbial source tracking data set, but this project is going to give us a chance to really pull all the data that's relevant together and hopefully come up with a much better picture. Maybe even be able to start making plans moving forward. So I think next slide, maybe Ruth will talk about.
Yeah, so one of the things that we're going to do, we'll be collecting the new data that Dr. Kilreese talked about and putting those data together with environmental attributes. And then like Dr. Hein said, we'll be collecting also previously collected data and sort of compiling all this together, and hopefully having a student help us with just compiling existing microbial indicator data, any source tracking data, and get information like what LaDon was talking about up front on ongoing projects that we can also dovetail with. And then we really want to use this in our project as a platform for facilitating bigger picture data sharing. So sharing, and disseminating data from our project, but also once we've compiled all of these other data, providing one location for people to get all of these resources. And we're going to leverage an existing website. And many of you are probably familiar with this. Whoops, that just jumped on its own. Can we go back one? So many of you may have already worked with us on developing this. This was developed actually under a NOAA Restore project, but we have this wonderful website and this existing outreach program called Our Wastewater Footprint. And part of that is making these resources available. So if you haven't already visited our website, or if it's been a while since you've been there, I invite you to go or click on the QR code, and hopefully that'll take you there. But once you do, okay, now Brandi. You can get into our resources section on that page, and then under data resources, you can find information like this that we've already collected from, to have existing data sources available. Our intent then is to add on to this and build in all the stuff that we've compiled, as well as our new data, and make it available here, so we can pull that all together in a holistic way and make it available to you all through this existing resource. So thanks.
Right. And lastly, we wanted to make sure this is something that has a legacy, that we're not just doing a one-off study, that we like to make eDNA accessible to those that want to incorporate that into, or at least understand it maybe into their data streams. At one time, everybody was just doing, the classical microbiological techniques, and then we have been moving towards, qPCR, and I think that's a lot more commonplace now. A lot of labs, source tracking, the ones that are focused on source tracking are interested in source tracking. qPCR is becoming very common, or more common. And of course, it's, not as, technologically elusive as it used to be in terms of cost and in protocols. And what is on the horizon, and that has been over the past, decade even, but definitely in the past few years, as sequencing has become a lot cheaper, it's become a lot more accessible, that eDNA, the environmental DNA, that is, really complementing existing data sets. And I think could really take things to the next level by discerning multiple species at once. And so just in brief, we're taking things, at wastewater and oysters, and we're showing here cows, pigs, and the fowl, swine and fowl. And all of those that interact with that water in some way. It could be their feces, it could be their, shedding of their skin or their feathers or the material, or just, even living in that water, of course, like, oysters. And they do leave behind parts of their DNA, right? So, it's not just, the bacteria that's left behind, but even the organisms, or so those species themselves are leaving a signature behind. And that's, like I said, in their DNA. So taking that DNA and doing what we call metagenomics. So it's just sequencing all the DNA in the sample, extracting it and sequencing it in a technique called metabarcoding, something that you might have read about. Of course, you can do qPCR, which just targets one specific gene and gives us a quantity of that gene. But metabarcoding gives our sequencing, can give us all of the DNA. So we'd like to, distribute a protocol, a training, and also not in just how to collect it, but also how to, sequence it or get it sequenced. And then, of course, get it analyzed. So analyzing is a big part of that too. Having all of this data, all of these sequences aren't any good unless we know what to do with them. So we are, built in to this proposal, doing live and virtual, workshops, recording those workshops to also train users, and then creating a knowledge center and hosted on the DISL website, as Ruth has already pointed out. So we'd like to bring you guys together, not only during the stages of this project to help make this project your project as well, and to also keep you informed, but also train anyone that is interested in it at the end to use these tools. So that takes us to our sampling locations. These are these sites were strategically selected, and there is some modifications that can be made because, of course, if there are sites that you have been sampling for a long time, or have a legacy data set, or we'd love to leverage those other programs, and of course, other archived data sets. But we did select these 13 sites, and again, they're can do some modifications. We can't really, within our budget, add more sites, without, that would mean increasing our budget. So we'd like to, definitely have to stay with the 13. Our sample frequency is monthly, in that we are kind of, we've maximized the budget. We've eked as much as we can, out of it, but there is some room for modification. So these are the sites that were chosen. You can see that our sewage spills in 2020 and 2021 are marked on here as well. And our potential resources or sources of that we're interested in are also denoted on here. And this is a great working map. And I think you guys can, I'm happy to share this proposal. I'm happy to share this PowerPoint with anybody that wants it also at the end.
Hey, Brandi, before you go on, I just want to recognize Baykeeper for sharing the data for the sewage spills, because that came from their website and their offices, and they have some cool interactive information on their website that people should check out. And also recognize Elizabeth Heap for making this dream of a map a reality for us for the proposal.
Very true. Thank you so much. I appreciate you you saying that too. And yes, we talked about it, but Elizabeth ran with it, and this has been very useful. And you can see the watershed too, that like this. I mean, I know they call this America's Amazon, but it really does illustrate just how much of the watershed that Mobile Bay takes in. Of course, we're interested in for this particular project, just what's happening in these potential impacts to the Mobile Bay water quality. But of course, this is fed from a large portion of of Alabama, including Georgia and Mississippi. This is a little bit of an overview from what we just talked about, microbial source tracking 3 ways: the classical, biogeochemistry, and some of the advanced techniques. And then, of course, the environmental attributes that are going to come from our own YSI monitoring, NOAA buoys, USGS gauging stations, and so forth. The timeline is we've heard from our program manager that April 1st is when they expect to complete the onboarding for this proposal. They've been working on through they have the same challenges that we all have with COVID and of course staffing. So it's a little delayed from what they had hoped, but they're on track for beginning of April. We're recruiting students or technicians to work on this project now, which we have not hired anybody. So if there's somebody that you have in mind that is interested in doing this, that would be great. Please send them our way. Our application period is still open for accepting graduate students, and of course can accept technicians anytime. We are hoping to compile the existing data, complete that this summer, and do the things that Ruth was mentioning, and get those online and get those available to to our partners. We hope to get some preliminary field samples done this summer as well, but our dedicated field work as proposed will be 2023 and 2024. So this first year will be really trying to get off the ground and get everything together. The working group meetings, which includes you all biannually, and of course the eDNA toolkit that I described is anticipated for 2024. Of course, in addition to coming to these stakeholder meetings, we really do want your feedback. You can do that now. You can give us some feedback now, or if you feel more comfortable, send us an email, set up meetings one-on-one. That would be great. However, you want to let us know things like sharing existing data and existing project information. We'd like to know that. We certainly aren't intending on stepping on anyone's toes, but I think we could just really work together to make this data set and other data sets stronger. We'd love to hear from you about refining these sites, and I'm going to go back to that site map in just a moment. And also, are there additional samples that you could collect or we could collect and we can archive for future? We have plenty of -80 space that we can go ahead and get those samples, and we may not be able to sequence right away, but we can certainly get them archived, and we can talk about ways that we can incorporate and combine sample sets or data sets. Feel free to tell your colleagues, feel free to invite your colleagues. That is absolutely fine. This is not a closed door or a private project in any way. And then if there's ways that you might use the data, we'd love to hear about that too. And also what products you might have. I will go back to the methodology too, and the approaches. Are there things that we're missing? Data types that we're missing or analyses that we're missing that we can add. Maybe we can incorporate your data sets as well. And so with that, I'm going to slide back over to the sampling locations, but are there any questions?
Brandi, we did have one from LaDawn. Do you want to just go ahead and ask your question, LaDawn?
Thank you, Ruth. Yes, I think I heard, I'm not sure who said it, but maybe birds are a minor contribution. Are you going to be any of these sample sites near bird rookeries or other areas that concentrate birds?
Yes. Let's see. So we do have some. So
Gaillard.
Well, the site that we specifically picked with birds in mind and your shellfish was site number 2 down there with the little bird in Mississippi Sound, which is near that little bird island.
Okay, good.
But yeah, I mean, we could have bird influences in a variety of places, but that was one that we put in there, especially to be like a hypothesized bird positive.
Yeah, I missed that icon. So thanks. That answered my question. I did have one question. Our project was funded mainly to try to inform public health. Or is public health involved in this? I know they're really busy. So they certainly would be a great end user of this information.
That's a great.
I believe we invited Byron. I don't remember what came of that, but we will definitely circle back with them. That's a really great point, LaDawn.
Just to add to that too, I know the issue Baykeeper dealt with forever was the duck pond in Fairhope being a significant contributor, whether it was or was not a significant contributor to water quality along the Fairhope beach in that area. So that may be another site you want to look for bird populations, the ducks, which are actually geese mostly. And then the other one, I agree with LaDawn about partners and was wondering whether or not y'all considered including sewage treatment facilities like Moz or Fairhope or any of the other ones, as well as like Mobile County Public Health or Baldwin County Public Health, just to see if they should be in this mix at all. But
that's a really great suggestion. I didn't even think about directly inviting the wastewater treatment plant folks, but we can definitely do that. We can loop them in. And one of our sites is close to, I forget exactly where, so 10 is close to, is the Fairhope site. And it was picked for that reason. There is a wastewater treatment plant outfall there. I didn't even think about that, the duck issue. I remember that from a few years back, especially getting a lot of attention. And it's right there. I mean, it's close to the wastewater treatment plant as well. So,
and depending upon who you talk to, they will say it was the ducks and then it was the cats that chased the ducks. So it's a wildly entertaining, and you should cocktails and I'll tell you fun stories.
But I think that'd be a great spot. I'm glad you brought that up, because I think that may be a very good spot where we can, Brandi and Sinead can do their magic and we can parse that out.
Yeah, I think so. I think having those, as you mentioned earlier, Ruth, those positive, known positive sites like the bird site, and I think that's very that's important, especially when it comes to eDNA, to know what that signature is, because the more we get, and we talked about having sites in the middle of Mobile Bay, but we decided against that because that's just going to be very diluted when we're looking at some of these signatures. We do have 1.5 that we're using as a control site. That's going to be a bit more of the diluted signature. We might, yeah, so having those positive sites, those is also very important. So being near those, sewage treatment plants, wastewater treatment plants is important.
Melissa also suggested, that we can get pretreatment samples from the wastewater treatment plants. And that actually is something we talked about and plan to do as part of our preliminary, like you were saying, kind of get the baseline for what we should expect to see in the environment. And the cool thing is we've been working with Daphne, Fairhope, Mobile, and Dauphin Island wastewater treatments, as well as over into Mississippi and Florida. And they are great. They have been great and have always allowed us to come in and get grab samples or get 24-hour samples. So I'm glad you brought that up, Melissa. And I think we will definitely take advantage of that.
Cassie has her hand raised.
Yeah, so just looking at the locations for sampling on the map, that's really supportive of what we've been seeing. We're really interested in Dog River, the Halls Mill Creek area, Fish River. Those are sites through our testing that frequently have high bacteria results, and we're really interested in learning more about that. Another location that I know you said the 13 sites are pretty set in stone, but that we've had really big interest in is Chickasaw Creek as well, through high testings we found there as well, just for future reference. But I'll talk more later as Cade wanted to join in, but we'll definitely want to touch base with you on how we can help sampling in those areas.
I think it was budgetary wise, because we planned our budget on a per sample basis, and so I think it's the number that we're really married to. And so if we want to move things around a little bit, we can kind of do that. But, then we start risking going over in budget. And everyone here knows about budget. Don't need to.
We did take into consideration, Cassie, that hopefully will help is that you'll notice we tried to pick sort of the mouths of those waters, so the mouth of Dog River, and so we're hoping that we can pick up stuff, sort of treat that mix of essentially non-point sources, but treat the rivers themselves as sources. And so we're grabbing a lot with that one sample. But we had talked to Cade, and hopefully with you specifically, we can work with your student groups that are going up in there and sampling, and we'll be able to, and we also put this in the proposal, even if we don't have the funding in this project right now to run every sample, we can archive samples. So we can take some samples. So if you guys have other people who are collecting data and we want to just bank it for future, we can provide that resource as well. So Sinead, did you want to jump in there too?
Yeah, just to reiterate what Ruth just said is that, budget-wise, we're limited in what we can do, but it's not that costly if baykeepers going out and sampling at sites that we don't have the map right now to filter the water and archive those samples for later. We wouldn't have, real-time or immediate numbers, but it might be very interesting to build out some of those kind of supplemental collections where if an opportunity arose and we could do a retrospective look where we could compare the measurements we're collecting right now to some of these more molecular measures that will allow us maybe to fill in some of the gaps. The strengths of the classical methods and the qPCR methods is that they're telling us about things we know about. And I think the beauty of adding the eDNA, that is Brandi's expertise, is that it's going to allow us to identify undefined contributors. A lot of our tools right now just let us look for what we know to look for. But if we can archive samples, we'll be able to, go back and really broaden our understanding of what's going on in these systems.
And Kim Cressman also had a question, I think getting at differences, for example, between septic systems and wastewater treatment plants. It says, do you have any sites near primary septic areas such as residential areas on the map? I can partially answer this, that we have some ability to distinguish, because in a lot of ways, especially coming out of these rivers, and we've done this similar work in Fowl River, and I'll share with you guys a couple of publications on this, but we can use isotopes. So one of the great things about nitrogen isotopes is we can get at distinguishing raw sewage from processed sewage, and essentially a septic system is more processed than raw sewage. So we can get at some of that. And I think what's going to be cool is putting that information that's a little bit more circumstantial together with the information that Brandi and Sinead have, can help us get at some of that. But I'm going to turn that over to you two to talk more about what you can do.
Sinead, do you want to go, or should I?
Well, I think it's just, the molecular tools, right? The qPCR, you have to know what you're looking for ahead of time. The eDNA, we're basically looking at the signatures of everything that's passed through that water body. And so for some of the, say, fecal contributors that are challenging to look for right now, like feral hogs, the eDNA may actually give us a really good tool to complement the tools that aren't necessarily as robust, say, as looking for human contributors or ruminants and so on. And so I think it was one of the strengths of the proposal was incorporating kind of the classical tools, the current state-of-the-art molecular tools, and essentially taking a step into the future molecular tools in terms of how they're applied to water quality.
Yes,
I think, Kim, this is really one of those situations where all of our different lines of information are going to come together to help put that puzzle together and get at some of those different signatures.
Right. So specifically, the ones that we proposed were Bacteroidetes, which is the total, just how many are there using the 16S rRNA, human associated, which has a distinct, genetic signature, cattle associated, waterfowl associated, and feral swine. Now, something that Sinead mentioned is that eDNA gives a signature of everything that has interacted or passed through that water body, which is, very important in that it could be currently there, say oysters that are, currently residing in that water, or, the, wastewater that is currently being deposited, but also could have, like Sinead mentioned, passed through. So eDNA typically sticks around. Estimates are currently about, a few weeks to a month. And it's a little hard to say because, of course, it's dependent on, the water movement itself. It's dependent on, stratification, breakdown of that DNA, which different times of year, DNA can break down more slowly, be more preserved, for a longer period of time. And likewise, it can also, recycle more quickly in the environment. So, of course, eDNA, as it is a future step in research, we still need to understand more of it. And I think that's just the strength of this, is that we have those that we've been using for decades, those microbial indicators and isotopes and nutrients. The qPCR that has, that has been coming on, and, we have set protocols that are used in, a number of labs across the nation. And then eDNA, which, adds a bit more of a exploratory, understanding to these systems.
So we have several questions that have popped up in the chat. Melissa asks, have you thought about adaptively managing your site? So refocusing resources in areas that seem more or less promising as the project goes along, is how I'm interpreting that.
Certainly, and that's absolutely something great to bring up. Yeah, off the top of my head, I mean, I think that's not a bad idea. I think it's good. I mean, if you find a site that it's, you know, not impacted, that we thought it was, you know, do we shift over to another site? I don't see why that couldn't be, you know, something that, in the reason why we're having these biannual working group meetings. I think that's something very important for everyone here to, you know, for us to talk about. Ruth or Sinead?
Yeah, I would say it's kind of a, you know, yeah, we'll obviously be doing that as things go along. But I would say too, though, there's some value to seeing, you know, where the signal is and where it isn't. But that they're, you know, I think we're going to be assessing the data as they come in and adapting accordingly.
And we are planning, like Brandi said, you know, we're going to be doing some hopefully preliminary sampling this summer, and that'll help us nail down some of these ideas, because you never know what you're actually going to get until you get out there and start taking samples.
Another thing that just kind of came to mind too is I'm glad you asked this question, because it's you know, forcing us to think, is that, you know, it may not be you know, may not appear to be impacted at a certain time of year, but over the course of a year, it might turn out to be important, and we don't know whether that is or not, unless we completed it to fully. This is a 3-year proposal. It's a 3-year project. So I would love to see this live on, and we're definitely going to look for other sources of funding to, you know, continue this kind of important data gathering. But yeah, that's an excellent question. It is definitely hard to set the sites without, as I'm just reading your comment, thank you, without understanding, you know, that preliminary data first. So, all right, thinking about prioritizing areas for resource allocation, but also values of the negatives. Yeah, and a negative in, you know, wintertime might be, you know, not in the summertime, and vice versa. So yeah, don't you wish we had $10 million?
I think Missy also had a question about the validation of the qPCR results.
So what I was going to ask about that is, really, we know that there can be some mixed contributions in watershed, and I'm thinking specifically associated with whatever primer you're using for, you know, cattle associated or something like that. We know that sometimes ruminants crosstalk in those qPCR results, and I wasn't sure if you were actually going to go out there and do some fecal sampling or something like that, some validations to make sure that what we're calling cattle associated or whatever organism associated, that it's not interacting with something that's very similar.
So we've done mostly, we haven't done any fecal collection to validate the primers. We've focused so far in the work in my lab, we've worked on just the totals and the human associated. I think we'll, once we start sampling, we may do some simple validation. There's a previous study that used these same primers over in Fairhope. And so, you know, the short answer is I don't know right now. We're going to we'll see what the data look like and if they make sense, and then we'll probably have to do a little bit of validation. Yeah.
And we are actually, I mean, Dr. Bloodgood can tell you how fun it is to go out and get wild boar fecal samples. So we have already started collecting some of those, because those have been the real wild card, as it were. That's kind of that's a pun. But, you know, because we didn't have previous work really on the wild boar here. So yeah, these are all excellent points. I think that's all the questions that were in the chat. Does anyone have other questions?
If somebody has access to, in the case of the wild boar, I know we were working with Grand Bay National Estuarine Research Reserve to try to get some captures there. And if there's others that can help us, you know, capture some positives, you know, that would be great.
So I've been jotting down all of the suggestions as we've been going on. These are fantastic. LaDon, I know we've talked about, just a quick recap while you guys are thinking about if you have other questions. You know, LaDon, you talked about the ongoing Sea Grant project. We'll definitely follow up on that. Fred, also thank you for putting the link in for your data into the chat. If anybody else wants to grab that, I encourage you to do that. Scott Reichard also recommended that we reach out to Dr. Tarnecki, who is the new faculty at the Shellfish, who's working with the Shellfish Lab through Auburn. And then we will invite her in. We will definitely circle back with public health and make sure that we have someone or someones from public health participating, thinking about the duck contributions at Fairhope in addition to the wastewater treatment plant, also reaching out to the wastewater treatment facilities and, you know, getting those folks involved as stakeholders, as well as getting those grab samples, which will be nice, you know, essentially those positive endpoints for us. Keeping in touch with Baykeeper, you and Kate and Cassie, to collaborate on the Chickasaw Creek sampling, and maybe taking advantage of your student workers, and also for anybody just working with us to bank samples. If you have ideas for collecting samples that we can then bank, like Sinead talked about, thinking about adaptive management of the sites and refinement of the sites with your input, you know, as well as from our preliminary sampling, and then also getting some of those ground truth samples from the cattle and ongoing with the wild boar. And then I think that's what I have, unless I missed something.
I heard mention of Patty Savecki as well. Had a proposal. Would it be appropriate if we reach out to her to start a conversation? I can't remember who mentioned it.
That was what LaDon. That was
Okay.
what LaDon had suggested, and she's reached out to me previously for work that we did on Fowl River. So I think that would be fine. I think that would be great. We also have some additional comments in the chat that we could reach out to wildlife control and recreational hunters for this area, for the culling of the hogs for samples. And that's kind of what we've been doing over in on the other shore. So we could potentially do that over in Baldwin. And then Fred adds, in addition to the beach monitoring program samples, ADEM collects Enterococcus samples for coastal water quality monitoring. And Casey mentions that SWCC has an active feral swine project in Baldwin and Escambia. That would be great, Casey, if you don't mind getting us the contact information. And Elizabeth too, if you know of the contact information, want to share that along, that would be fantastic, or we can look it up.
Yes, Ronald is also asking, are you collecting more than 100 mils or are you doing more water? You mean collecting more than 100 mils of water? Yes. As for eDNA, we're going to collect, hopefully, as it takes a lot of, it does take a large sample volume. So 5-10 liters, and, you know, try to collect as much as possible and filter as much as possible. It might be multiple filters that will have to be extracted and combined for ultimately for, you know, sequencing. And we're going to use that large volume, right, ultrafiltration So using Steravex and extracting those, and then we just won't be able to get enough through a flat filter. So Steravex, Steravex is an accordion style filter. It's in a cylinder, and it looks, it's all folded up like an accordion, just kind of a lay term for it. And so you can get a lot more, a lot more water through there. And we'll try to use the same filters and the same extractions that we pool for qPCR and for eDNA. And then, so does that answer your question, Ronald? Yep, and then 500 mils for, yes, is what Sinead has been collecting. When we get to eDNA with the large, because it can be very dilute, right? These the signature of these particular species. So we 500 mils is great, but we try to get just as much as possible, just so we can really get see those signatures clearly. Thank you, Casey, for the for Jim Hartman's contact information.
Oh, great. Thank you.
Thank you all for coming. We really appreciate it. And thank you for taking the time. I know we've gone over a little bit, but we do appreciate everybody's contribution. We anticipate another meeting later on this year after we've gotten through some preliminary sampling. We're going to reach out to some of you that you've mentioned about refining some of these sites. If you think of something later, feel free to email Ruth or myself or Sinead. That's absolutely fine. Just let us know what you think, what we can add. I did talk about budget, but we're very flexible. We want this to be a true community effort. So we're just very grateful for everyone being here today.
Yes, definitely. Thank you.
Cassie raised hand just now, or was that from earlier?
I think that was from earlier.
Okay.
Sorry.
No, that's all right. I was having a hard time monitoring both screens. All right. Thank you, everyone. Stay well, and we will be in touch very soon.
And Sinead and Brandi, if you want to stand for just a second, I can share some things with you real quick.
Great.
Yeah. Thanks, everybody.
Thanks.
Thank you.
Bye.